I'm Harry Glurkian, and this is Moneyball Medicine, the interview podcast where we meet researchers, entrepreneurs, and physicians who are using the power of data to improve patient health and make healthcare delivery more efficient. You can think of each episode as a new chapter in the never-ending audio version of my 2017 book, Moneyball Medicine, Thriving in the New Data-Driven Healthcare Market. All right, Richard, welcome to the show.
Thanks, Harry. It's great to be here.
Richard, I was putting together my notes on all the different things you've done, and I'm like, oh my God, I feel like I haven't done anything with my life relative to what you've accomplished. I mean, you started out as a nuclear engineer, but then you make this complete turn into biological world, and I'm making that assumption. I think you said somewhere you read a book. I can't remember which book it was.
That's right.
That totally flipped you into that direction, and then it was bioinformatics, protein engineering, and then now gene editing. Tell me a little bit about the path here.
How did I get there? Yeah, no,
Yeah.
That's right. It was a meandering path, especially early on, but then the last, I'd say, couple decades, it's been pretty consistently in the field of biotechnology, especially protein engineering, and now metabolic strain engineering. We'll talk a good bit, I'm sure, today. But yeah, I guess sort of to rewind, you're right. I did study nuclear engineering in college, and I was working for the U.S. Navy actually as an analyst, civilian. And I was on a ship actually out in the middle of the Pacific Ocean, and I had just been sort of spending my time reading a book called The Selfish Gene by Richard Dawkins. And it completely transformed the way I thought about the world, my place in the world, how evolution worked. And I was completely smitten with the concept of evolution and what it could do, the complexity that it could craft, that nature has done over billions of years. And from that moment on, I had a deep interest in evolutionary biology and the principles of that really elegant algorithm to optimize exceedingly complex systems. So it wasn't long after that I found myself ultimately, working for a biotechnology company to be able to practice some of the principles of evolution, although at a much smaller scale, at least initially, with proteins. So.
Yeah, it's funny when you said the elegant algorithm, and I'm like, wow, I wonder, there's got to be a lot of them, right? If you think about, evolutionary biology. But I think the company you're talking about is Codexis, was the one that you went to?
That's right.
And so
actually, yep, that's right. Sorry.
You worked on protein engineering, drug design, but relying on bioinformatics, statistical analysis, machine learning, evolutionary programming, sort of picking all those things together. How did you bootstrap yourself into a position where you understood all of these different components?
That's a great question. I think it's like a lot of folks who take an interest in something. It's, my career has pretty much been dominated by being interested in things and being passionate and being curious. And so those led to all of the experiences that I've had, really. That's the short answer. It was driven by this interest in biology, but because I had in my studies in nuclear engineering that we talked about earlier, I had pretty much always worked on the computational side of things. I was not good in the lab. I'll never be good in the lab. I'm always amazed at what the scientists who can actually generate the data that I get to play with can do. It's stunning. So, but I get to sit and I get to play with that data, and I, for many, many years, written software and algorithms to process that kind of data. And so that sort of was a natural fit for me.
So just so people can sort of get an evolution of where you were, and because we're eventually going to get to where you are now, but so sort of, what was the main focus of Codexis, or the special sauce?
Yeah, great question. So actually, Codexis was a spin out from a company called Maxigen, and Maxigen started, if I remember correctly, in the mid 90s. The inventor of the technology, a gentleman by the name of Pim Stemmer, was really one of the pioneers in the field called directed evolution, which ultimately went on to receive a Nobel Prize. Francis Arnold won in 2018 for that field. Pim Stammer was one of the great luminaries in the field early on. And he had developed this technology that would allow you to do evolution in vitro in the lab, primarily around smaller sequences, evolving genes, proteins, enzymes. And so that core technology was called DNA shuffling. And it was very much all the main principles of evolution. So mutation, recombination, selection, all was being carried out in vitro, very high throughput, very fast to evolve proteins and enzymes for different properties. And so Maxygen, the founding company in the mid-90s, got started. And then in the early 2000s, took that core technology and licensed it out to different subsidiaries. And the one that I was associated with, I started with Maxygen, but then I went with the Codexis subsidiary. And they use that DNA shuffling technology to work with enzymes primarily for pharmaceutical manufacturing processes.
But you invented, I think it was called ProSAR while you were there. It was sort of to sort through, protein mutation faster. Was that like, should we have seen that as foreshadowing to where you've sort of progressed to and where you are today?
Yeah, I think so. Actually, it's interesting. Before I had even joined Maxygen, so it turns out my wife was a scientist at Maxygen. When we first started dating, she told me about this really interesting company that she worked for. And she described, it was evolution in the lab. And I was, of course, already keenly interested in evolution, as we talked about earlier. And I think to this day, my wife wonders whether I married her for directed evolution or of course, I married her for the wonderful person that she is. But I very clearly remember before I even came to Maxygen, given the background I had in software and algorithms, I understood what they were doing in the lab, at least as much as my wife could describe it. And like most people with the background that I have, statistics and optimization it's sort of a natural, it's sort of an obvious thing that you would want to do is given genotype and phenotype data, how can you search through that space more efficiently by trying to model the system? This is something that, statisticians have done, for, decades. And it was just sort of being in the right place at the right time. So when I then went to Maxygen, I and others, were very interested in applying these principles to the searching and sequence space.
Yeah, and if you think about, computational capabilities, that whole space has changed, dramatically compared to what we could do in the 90s.
Yeah.
And then you went on to, if I've got my history correctly, Inscripta.
That's right. Yeah.
And tell the world a little bit about Inscripta, because I'm not sure that it's, how well it's understood or how well the company is known.
Yeah, so Inscripta is a life science tools company. So the easiest way to think about it is they want to do for writing what Illumina has done for reading. So they want to be able to, at scale, intervene in the genome across, and initially they're working in microbes, eventually they'll be having mammalian capabilities as well. But they want to be able to interrogate the genome at scale. They want to offer tools, benchtop tools, and reagents and software to be able to essentially automate and scale up as much as possible the editing process so that researchers can focus on their research goals and questions, which is if I intervene here and there and everywhere, as in the case may be with these capabilities, and then being able to test or phenotype, what the result of those interventions are, it's hard to overstate how transformational that is, right? I mean, genome biology for years has more or less been dominated as an observational science. With this ability to go and intervene at scale, you're really, for the first time ever, turning it into an interventional science where you can really get at causality by making the changes in the genome rather than just reading them passively, the
Well,
sequences.
yeah, I mean, if you can make the changes you want on human, the industrial application is unbelievable. I mean, the things that you could, design and then have that produce something that is for some particular downstream use would be incredible. Now, but you guys weren't using, I think you guys said you decided not to use the Cas9 approach. You used something called, I think it was MAD7. So what was the motivation behind that?
Yeah, so it's still straight up CRISPR. It's with a different nuclease. So early on, Inscripta was looking at how to enable this high throughput, massively parallel editing capability. The inventor of the technology, his name is Andrew Garst. He was a co-founder of Inscripta, and now he's actually a co-founder of the new company that I've started with him and two other gentlemen. And that core technology to be able to do high throughput CRISPR is based on the standard editing technology that centrally involves a nuclease. And Inscripta early on was looking at the landscape around licensing and enabling researchers. And because of some of the issues around licensing of the nucleases that were out there, Inscripta made a concerted effort to go and discover and develop a different nuclease. It would still do the basic process of finding DNA and cutting it in the right place. But the advantage of using this other enzyme is that Inscripta offers it basically free to the world to use, so that they're not encumbered by some of the more onerous licensing terms that are out there for other nucleases.
So just so everybody kind of understands Inscripta, like what was the process, let's say, before Inscripta, and then now if you utilize something like the Inscripta platform?
Great question. So CRISPR works, it's amazing, and it well deserved the Nobel Prize in 2020. It is truly stunning, its precision and its efficiency, but it's still fairly low throughput. So if you want to go in and you want to make a change to a genome, you have to design your sequences so that they are targeted to the right location, and then the nuclease performs the cut, and then there's some repair process to usually insert the sequence that you want. And that can be done by hand, manually. That design process can be scaled up, obviously, with computational tools, but you'd still be limited physically to doing only a small number of changes. It's just the molecular biology associated with bringing all the right reagents together is sort of can be a laborious process. If you're making 1 or 5 or 10 changes, that's not too bad. But if you want to make hundreds, thousands, tens of thousands, that's a different proposition. Yeah.
I was just thinking, even doing 1 or 10, and doing them right, and then now you're talking about hundreds or thousands and doing them right. It's a completely different order. I mean, so if that's what Inscripta does, then I almost, answer my own question of your data sciences group to plan out what you're going to do has got to be, very good. I mean, your analytics capability, is that what you spend the majority of your time working on and thinking about?
For sure. When I was at Inscripta, that was the majority of what the team and I did, was to think about planning the experiments. And then, ultimately when the data come back, you have, when you look at all the data coming back, you basically have millions of data points. When you multiply, all of the sequencing data that comes back by the number of conditions and the number of edits that you have all across the system, you're processing large, sets of data to understand what each of these edits do. Mm-hmm.
So, if I had to ask you, so you've seen a lot, you've sort of made this evolution, and I want to get to Infinome in a moment here, but if you had to summarize sort of the impact of the computational methods that you've worked on the biopharmaceutical industry, how would you sort of put that into context?
I mean, it's hard to overstate the importance of computational tools. I mean, you couldn't do much of this work without that. Certainly on the informatics side of things, just managing the data, it's not that sexy, but it's of course critical.
Yeah.
And then once you have all that data, actually turning it into meaningful insights it's profound. The algorithms for evolution do work though. And so one of the interesting things is DNA shuffling technology that we talked about earlier worked without really a lot of informatics. You would basically apply, survival of the fittest to molecules and it would work. And actually quite well, but it was ultimately a blind process at the end of the day. And so to accelerate the fitness gain, you want to try to make use of that data to drive towards higher levels of performance in your system. And that really you can only do when you start interrogating what we call the genotype phenotype map or relationship. And that's allowed us to accelerate the process of evolution more than ever before.
So that makes me ask the question of, is that what you sort of learned at Inscripta that guided you to start Infinome? Or was there other pieces of the puzzle that sort of the light bulb went on and you're like, I need to go and start this next entity?
Yeah, that's a great question. So actually all of that sort of statistical modeling, people call it machine learning now, was done quite a while ago when I was back at Codexis. And to really understand the history, what happened was that we had developed a lot of these capabilities, but at the gene level, engineering enzymes rapidly. So using statistical modeling, high throughput automation, software and information systems, and also a suite of sort of concepts about how to generate the data, plan your experiments, and best move quickly through the cycle, the design, build, test, learn cycle. All of that was very well developed while I and my colleagues were at Maxgen and Codexis, going back a decade or more. And so it was really around 2010, 2011, where that technology for doing gene-based rapid evolution had evolved quite a bit. It still had room to grow, and Codexis is now arguably the state-of-the-art protein engineering company in the world. But what we were experiencing was a desire to move up to larger sequence spaces. So moving beyond just a single gene, we wanted to move to pathways and genomes, because we believe the bioeconomy is in many cases going to involve, engineering whole genomes.
Right.
And we were very excited at the prospects of being able to do this. And we had a strategy, we had a playbook, because we had developed it to do single gene evolution, or maybe a couple genes at a time. But what we were missing, Harry, and this is where kind of to complete the circle with Inscripta comes in, is what we were missing for many years was the tools to be able to go in and make those changes across the genome. As it happens, working with genes is fairly straightforward and has been for about 20 years. You can go in and diversify a gene very easily, very cost effectively. You can make all the single nucleotide or amino acid variants that you care to make, and then evaluate those through high throughput systems. You needed something like that ability to make those sequence changes, but at the pathway and genome level. And that's what was missing for almost a decade. We were waiting to apply this strategy, but we didn't have the tool. And so that's where Inscripta really came in, is that about 3-ish years ago, I was very fortunate enough to get hooked up with the folks at the early stage Inscripta, who was looking around at what to do with this massively parallel editing technology. And it was music to my ears and some of my colleagues, is that, oh, now finally we can go after the whole genome the way we've gone after genes. Yep.
Yeah, it's sort of interesting that you can dial it up and then have these changes happen. I mean, if I think back from where I started, like that was I don't even know if it was a dream. It wasn't even even a concept when you think about it. And it's profound and scary sort of all at the same time, depending on who's playing with it. But so now that brings me to Infinome, right? So you went from, this protein engineering company that's top in its field to Inscripta that seems like, and correct me if I'm wrong, that's working more on industrial applications of making changes to a bacterial genome or yeast or something like that. And now you're at Infinome. And okay, for everybody listening, including myself, what is Infinome? What's it going to do? And how's it going to change the world?
Yeah, so Inscripta is amazing. The technology that they built and will be offering to the world is just transformational. You simply can't overstate how powerful it is. And it's more than just industrial applications, although plenty of biotechs, large and small, are very excited about the technology. It also has a lot of application in basic science, antibiotic resistance, and all kinds of things that the academic community can dream up using this technology. There's lots of applications. So all that's fantastic. What's particularly a challenge on industrial side of the equation is that as amazing as the Inscripta platform is, it's like any other technology stack. It's one piece of the puzzle. It's very important. It's critical in many ways, but it's not sufficient to do rapid genome engineering all by itself. What you find, and it's also true, going back to days at Maxgen and Codexis, is that the core, DNA shuffling technology, and then ProSAR later and so forth, all really important pieces of the technology stack. What we found, because we were part of developing the whole ecosystem, is that you needed everything else to work together almost seamlessly to be able to run very quickly through the whole process. And so what Infinome is doing is it's certainly going to use the Inscripta technology as a core part of its stack, but then we bring together a host of other capabilities and experience or expertise to be able to run this in the synthetic biology world, the famous design, build, test, learn cycle, very efficiently, very cost effectively.
So is this a service? Because Inscripta is, a product per se, right? That might be sold to someone, but is Infinome more of a service of doing it because of all the different pieces that need to come together? Or can I buy this in a box?
Yeah, no, it's more of it's more than a service, I would say. It's a group of individuals with capabilities, wet lab expertise, informatics expertise, the know-how to pull it all together. It's definitely an execution team and a suite of capabilities. It's not an off-the-shelf offering, not by any means. Yeah.
So what do you say as assuming all of this comes together the right way, what if you describe to someone, what could you do? What would it be?
Yeah. So it turns out there's all kinds of opportunities in the bioeconomy that are just waiting for folks to go after, but they don't have the capabilities to be able to execute on them. So the Inscripta technology is important, statistical analysis, high throughput automation, all these things, are important, but very few organizations have been able to pull them all together in a way that allows you to run very fast, very cost effectively. And when you can bring that execution, sort of an activation energy barrier, if you think about it that way, you bring that down, now a whole suite of bioeconomy type applications are now on the table. So certainly producing bioproducts, proteins and small molecules that are high value or are commodity for that matter, they're now all things that you can go after, because it doesn't take 20, 30 people in 10 years anymore, like the way it used to engineer microbes. It's very typical over the last 10 or 20 years for large engineering efforts that took many, many man years, potentially hundreds of man years, many tens of millions, if not hundreds of millions of dollars to generate these biological solutions. Now we're able to do at a fraction of that sort of time and cost with the capabilities that Infinome will.
I mean, it sounds like, though, and I always go through this debate of doing it for someone else versus doing it myself sort of thing of you almost should do all the work yourself and produce the product yourself. That seems like it's where it's going to garner the largest value.
Yeah, and actually that gets to Infinome's business model, which is we are indeed going down that road. So we are technologists. We love our technology, but at the end of the day, and I should have given you the background here, if it wasn't already obvious. Inscripta was amazing, great fun, wonderful people, some of the best colleagues I've ever had in my career. And yet what we found is that at the end of the day, we wanted to take this technology and apply it to actual applications. That's what
Right.
ultimately led to the formation of Infinome. And so we ultimately had the idea that we wanted to build this technology stack to be able to apply it to real applications. And as we looked around at how we wanted to build out Infinome, it's definitely a core part of our business. It's sort of our reason for existence at one level, but we're actually going to pursue some mix of both internal applications and working with partners, depending on how the opportunities kind of play out.
So I try to always in the show is get to like that intersection of the biology and the data, right? There's the Inscripta platform sounds like it helps you efficiently apply the biology and know where to apply the biology based on the data that the informatics platforms that feed it. The question is now in Infinome, how are you looking at balancing those two pieces, right? The data analytics at different points and getting the product you want in the end, or is it stringing together the right pieces of the puzzle to create something from end to end? I'm trying to wrap my head around these two concepts.
Yeah, the data analytics. So that's a really important question and piece to the ecosystem. So as we've talked about before, the ability to diversify sequences, whether it's at the gene or the pathway or the genome, is sort of step one. And especially in contexts where you're making multiple changes, this is when the informatics becomes really important, is when you have sequence variants where you're making multiple changes.
Yep.
Then there's a deconvolution process to say, oh, well, which, interventions or combinations of interventions are leading to the phenotype of interest.
Right.
And that's where the statistical modeling and machine learning really starts to be powerful. And so Infinome is in the process of generating lots of data, not with just single interventions, but multiple interventions. And that deconvolution process will be critical to sort of unmasking the genotype phenotype relationship around the particular trait or phenotype of interest. This is definitely something that's been done for many years at the gene level. It hasn't really been done at the genome level because, again, we lack the tools to make these things, these kinds of libraries. But now we have it, and so now we're off to the races again. So individual projects where you're looking at these relationships between genotype and phenotype certainly are amenable to this kind of statistical analysis. I think what's really interesting is to think about down the road how much of that landscape, that genotype phenotype relationship, how generalizable is that? What are sort of the rules of thumb or guiding principles that you can apply across many projects? Maybe some of them are related, maybe some are very different. What are kind of the patterns that over time, with enough data, can you start to give yourself an advantage, when the next opportunity comes in? Is there something that you've already learned from the data that you've generated and the models that you've created that you can apply to the future? This is the classic data network effect that we think biology, has long promised to have. But I think because we haven't had the tools to go in and actively intervene, we don't really know yet what the boundaries of that possibility are.
Yeah, I mean, there's always it always seems like when we get to enough that there is a finite number of options that present themselves depending on the model that you're looking at. And of course, I mean, maybe across different models there may be that rule set may be different, but I think finding one and basing something on, which is why everybody seems to find one and then never move off of it because they spent so much time figuring it out.
That's true.
So
Yeah.
where's the company right now in its process? Because I feel like it's in, I keep wanting to say stealth mode, but
Yeah.
uh, you know, where are you in the growth phase or the gestational phase?
Yeah, so we're still early days. We're a few months into this, and so we're talking to lots of potential partners and investors, and we're just about wrapping up our first round of funding, and we do have some partner projects that are spinning up, as well as getting to work on our internal projects. So we're going to be getting going here. We've been going in earnest, but we'll be a little bit more public here very shortly about Infinome, what we're up to.
And if you had to like describe a perfect project, I'm sure that when everybody came together, they're like, if we could do this, that would be right, as opposed to some amorphous description of what it was. If you had to put it into brass tacks for people listening, what would you describe to someone as an ideal project from start to finish?
Uh, yeah, that's a great question. I mean, it would involve at a high level, there's the scientific and then there's also the business. And I can sort of speak to both aspects. So within business, much of it's not controversial, right? You want to go after high value products, right? Things where, you know, the, you know, the, the economics around scaling the process are not so burdensome that, there's already, say, commodity solutions out there. You'd like to go after things that maybe are at a smaller scale and sell at a higher, unit cost. Not to say that commodity solutions aren't also, or opportunities aren't also on the table. Um, but that just comes down to techno-economic modeling and, where are the opportunities where you can get into the market and produce something better, faster, cheaper than something that's already out there. So those are kind of typical sort of business considerations. Um, on the scientific side of things, there's a lot of opportunities now with this technology that we're developing, that are putting things on the table that heretofore haven't really been a possibility. So in particular, the whole space of natural products is a really exciting one. So it turns out that a lot of people produce natural products in sort of exotic organisms because that's where they're initially discovered, and there's large gene clusters in these organisms, and they just work. And it's for lots of folks, the perception is that, well, you do what you can do with what you have.
Right.
That's what you were given. What's the old Rumsfeld?
Yeah.
You go
Yes.
to war
Yeah.
with the army you have, not the army you want. And I
Yes.
think part of it was based in some practical consideration around like, well, you spent all this time and effort to culture these exotic organisms and do a lot of fermentation, process development, and it's working, but it's not working well, but it's enough to, be economical. With today's technology, to be able to move large DNA sequences around, recode them and optimize them for different organisms. And now with the ability to, once you have a microbe with, say, a heterologous pathway, maybe even really large ones from these other organisms, maybe 10, 20, 30 genes in them. Now you can, with these high throughput, massively parallel gene editing capabilities and suite of supporting pieces of the technology stack, now you can move through these, pathway and genome sequence spaces much more rapidly than you ever could before. So the barrier that was sort of there before, which is, well, even if I can move the pathway over, it'd still take me 10 years to get the bug to perform at the level that's commercially viable.
Right.
Now you can see a path where if I can move these pathways over and work in much more engineerable systems, then I can, I could get to my commercial endpoint much, much faster than ever before. And this is not something that was possible before Inscripta and the Infinome, technology platforms.
Yeah, I can tell you, like, I mean, I remember we'd be working on a, particular pathway, and then, okay, we think we got it working, but let's see how it goes. And then you'd have to wait weeks to get some sort of result. And then, it's not as efficient as we wanted to. Let's go back to the drawing board. And it would take forever for that loop to keep going back and forth until you, and I still say, hopefully, get to the result you wanted to, because there was no guarantee that you were going to
Yes.
tweak it to get it to do what you wanted it to do.
Yeah. It's
Very
frustrating.
painful process.
Maddening. Yes.
Yeah, it is, because every time you feel like you've every scientist would tell you, I got it, I figured it out, I think we got it, I think we got it to do what we wanted to do. So if you took sort of, just so people listening can get sort of time frames, because I'm big on this, the difference between evolution and revolution is time.
Yes. Yes.
If you wait long enough, yeah, that change will happen.
Yeah,
But
sure.
right now, what I see is technology accelerating things, and the timescales are being collapsed, you know, at much tighter timelines.
Yes.
If you had to talk about where we were sort of in genome editing, and then put that into a timescale and talk about where we are now, how would you?
Yeah, it's a great question. So the core editing technology that Inscripta has developed is it's orders of magnitude more efficient. I mean, there's things you can do with the Inscripta platform that you just would never consider doing by hand. It would, to make 10,000 edits or more across a genome, to try to do that by hand, it would just be it wouldn't be feasible economically, manpower wise. So that ability to do massively parallel editing is sort of without a comparison. You just simply would try fewer things. And it would probably take you even more people with existing molecular biology techniques. So that's already one like several order of magnitude level of boost
Right.
in efficiency. And then, as we talked about earlier, as amazing as that is, even that's not sufficient, right? Because now you have all these variants.
Right.
Now you have to be very efficient in testing them.
Right.
And it turns out that that's also a bottleneck. And so even with some of the best, folks out there today practicing, genome engineering, you still find that the teams are fairly large and relatively slow when it comes to processing these variants. So, and this one's interesting because it's not that the technologies and the strategies don't exist to do it. It's just very rare to find the sort of the folks who can bring it all together with the right information systems, lean, smart automation. So to give you some numbers, for example, and I'll go back to sort of enzyme engineering back, 15, 20 years ago. Teams would be 10, 15 people. You would do one round of evolution maybe every couple months. And after a couple years or more, you get to, your endpoint. Now, state-of-the-art enzyme engineering teams are much smaller, 2 to 4 people, one round every 2 weeks, maybe a month in the slower projects. And so you're already seeing multiple factors of speed up in the enzyme world. It's that same sort of step up that we're looking to do with pathways and genome. So much smaller teams, maybe a quarter of the size or smaller, with much more diversity going into the pipeline, thousands, tens of thousands of things that you're testing. So when you multiply that out on a number of things tested per unit person, it's maybe 3 orders of magnitude more efficient.
And so if you said, so now I need a quarter of the people or a third of the people, let's say, I'm able to do more. What is driving that? Is it the data science side of it? I feel like a lot of the biology has been there already, but is it in the industrialization of the biology plus the data science?
It's both. I mean, it's definitely, as we talk, you couldn't do this before these high throughput, before this massively parallel editing technology was developed. You simply couldn't. So that was a key piece that sort of opened up the floodgates. But now a lot of it is managing what you create, both physically in the downstream test, software and information systems to manage all the data, and quickly and intelligently getting to the next round of prescribed experiments that you want to do. Without all those pieces, you simply would be, sort of hobbled in the overall, sort of cycle time, and how much functional gain or leaps in fitness you can affect at each round.
Okay. And then it's tweaking at every single one of those stages to make each one better or more efficient.
Yes, exactly. Yep. And sort of a key thing, it's sort of an obvious point, Harry, but it's interesting after all these years that it's not widely appreciated, is the following, which is in every step of design, build, test, learn, there's, to steal the term from electrical engineering, there's an impedance mismatch.
Right.
So between build and test, for example, historically, there can be widely divergent, throughputs for build or test. Sometimes you can only build a few things and you've got a really high throughput test, or vice versa. And so what we've seen, what we've personally experienced and been involved in innovating around, is to minimize as much as possible that impedance mismatch between every step of design, build, test, learn. You can make orders of magnitude improvement if you pay attention to those mismatches.
Yes, and I always think about it as whack-a-mole. I fix, I make one part of it better. The bottleneck just moves, right? It just moves where it is. And I don't know if I ever get to the whole thing is just moving at the pace I want it to, because ultimately there's only so many things you can pay attention to at the same time.
Yeah.
So,
yes.
So you're telling me that basically what might take me 3 or 4 years to do by historical or old methods now might take me 6 months to a year?
Yes, that's right. At a fraction of the resources as well. So it's not just how long it takes, it's integrating that resource burn over that period of time. Possibly a factor of 3 to 5, perhaps even more, integrated over a longer period of time. We're looking at much smaller teams, much more efficient use of resources, getting to the endpoint much more quickly.
So who is this disruptive to? Assuming we can do all of this, right? Who is this disruptive to in out there?
There are many sources of disruption. I guess one would be, depending on what you're going after, for products that are based on, say, the petroleum industry. If you could move those into bioproduction processes and replace those other sort of conventional sources of production, then it would be, those sort of old style petroleum-based producers. So they would be potentially disrupted by this. I think one way I like to think about it is that, it's a big world. And sometimes people ask, well, what is Infinome long-term, planning to do? And while we definitely want to create products and be successful, our view is that it's a big world out there and that there's so many opportunities to go after. We're excited, just sort of as scientists and members of the human race on planet Earth. We're very excited that long term these kinds of approaches will find wider adoption. Now that the tools are coming online, and if we can help be a part of sort of blazing the trail, there's a part of us that would be very fulfilled and satisfied if we could see this technology getting used in other areas as well. Long term, if we can help be a part of that process, either actively or passively, it's up for debate. But it's one of the business models we're considering, which is, as we get better and better at this and execute on multiple projects, both internal and external, eventually, if we can help the rest of the world in some way, as a template, possibly licensing technology, expertise, and so forth. Because as I say, there's no way that Infinome, even if it became, a huge company like Cargill or DSM or ADM, large manufacturing, even for them, the world's a big place, right?
Right.
So we're very interested in pushing the envelope, being successful on what we go after, and then ultimately hoping that, and being a part of, creating the ecosystem that the rest of the world can also use to go after the countless bioproducts that are going to be developed over the next 20, 30 years.
And it sounds like over time, as you're accumulating the data and understanding I make this change and these are the implications and this is what happens downstream. I mean, at some point it becomes much more data science than just wet chemistry at some point if you're focused in a couple of very discrete areas.
Yeah, I think that's right here. And that gets to this really interesting and unknown at this point area of how much can you generalize the process and the information that the models that you're learning, how generalizable are those to other parts of the genome? So I've already mentioned this sort of sequence function
Right.
landscape several times. It's a concept that's been around for almost 100 years now. If you think of, genotype as latitude and longitude, elevations, phenotype, if you think of nature having developed lots of, mountains and hills across this, very high dimensional sequence function landscape, a really interesting question is, if I'm climbing up this mountain for product A, if I go after product A prime and it's similar to A, arguably I can use some of the information or a lot of the information that I've developed already around product A to extrapolate to A prime. I think what we don't know yet is if you go for product B and it's near A, but it's somewhat distant, how much can you extrapolate from what you learned about A and A prime over to B? And this gets to, is it really in the cards that you can create a global sequence function landscape for all possible traits and phenotypes? That is a very tall order. I don't imagine that's going to happen in my lifetime. But some of the rules for navigating these spaces, I think, definitely are generalizable. But then it gets down to how close do the landscapes need to be similar to each other for you to leverage, what you've already, sort of learned about.
But at some point, right, you get to know A well enough that there's an informatics approach to it, and you know that it's going to work because you've worked with it so much. And then you get to know A prime, right? I understand the generalizable, which would be awesome. But even as you're moving down some of these product areas, somebody comes to you and say, can you make that tweak for me?
Yes.
It becomes a lot easier to make the tweak than when you first started trying to understand A well enough.
That's right. That's spot on here. That's exactly right. And so if you're working in like related product classes, then there's definitely huge value built up over, proprietary, data sets and modeling that you've generated. You can definitely leverage that and move much faster than if you were starting from scratch, for sure. Yep.
Yeah, I mean, it's funny, right? I always used to say that, I ran a consulting firm for a while, a strategy consulting, and I'd be like, the first customer that comes by, we're going to do our best, right? The fifth customer, man, they got such a good, insight and answer because there were 5 that we learned from and we knew exactly what was going to happen. And I look at this the same way, but with more solidified data pathways, understanding what changes cause what downstream. And now someone says, well, can you make this slight tweak for me? It's not starting from scratch. There's an informatics backend that sort of you can dial up and get what you want. And so the time scale of being able to do it would also be less.
It will also shrink. Yeah, that's right. That's right.
Well, all this sounds super exciting and super scary all at the same time, right? Because I can think of all the great stuff that can be done, but then I can also think of the easier and easier this technology gets, the more you worry about who's doing that work.
Yeah, that's a good question. And that one, so just so you know, Inscripta takes that along with a lot of, people who work in this field. So the gene synthesis providers have faced this, for many years, and they have taken that very seriously. So they screen for, nefarious, sequences or uses that could potentially be problematic. Inscripta is the same way, is that, you can't just order up whatever you want and create new pathogens, that there are pretty strong restrictions against doing that. So it'll be interesting to see going forward how companies like Inscripta and others will continue to stay ahead of this. I think it's very important for them to take an active role in this, and not, because the alternative is that the government would step in and legislate and create a lot of bureaucracy and slow down the science. And so I think the industry, it behooves them, all these tool providers and users, it behooves everyone to, try to do the right thing here. And so far, everything that we're seeing from Inscripta and other companies is that they are taking this seriously, and they're putting methods in place to prevent uses that could be dangerous.
Yeah, no, that's good. But it's interesting because this whole area that you and I are talking about, the implications are profound, and I'm not sure everybody fully. I'm not sure that most people appreciate how quickly the things have moved compared to where they were. I don't know. I want to say 10 years ago. I mean, 10 years ago feels like a lifetime. When you look at the level of change that's happened, across the board.
Yeah, it really is stunning. I mean, I remember being in Inscripta and seeing the first real large scale experiments that I was involved with, at least, and seeing that come out, and seeing that we were literally editing, 5,000-10,000 different genomes with things that we precisely designed and wanted to have integrated into the genome. It just was stunning. I couldn't believe that I was really looking at, the data that was really corresponding to reality out there, that we had created, 10,000 new organisms and something. I mean, in a precise way. People have been doing random mutagenesis for decades, but like in a directed, precise, conscious way, having that power, I'll never be able to describe it. It was something, as a computer guy, I had long wanted, because I can sit and write out sequences, at will. And I had always wanted this ability to do this for genes and pathways and genomes. And so to actually finally, hold it was really special.
It's funny because I've always said over the years, like biology always, you can come up with a great thing, you can map it out, you can do all the work you want. It doesn't mean that biology is going to participate willingly, right? And now what you're saying is we're getting a whole lot better at figuring out how to get the software of biology to do what we want it to, or manipulate the hardware within biology, however you want to look at it, but to get it to do what we want it to do when we want it to do it.
Yeah, I think that's right. And actually, we didn't really linger on this. I had talked a lot about my interest in evolution, but just to be very explicit about it, because it's important, is the reason why this technology is so important is because we don't know the rules of biology. If you knew the 10 or 20 changes that you needed to make, and you just went in and made them, and from first principles could design these biological systems, it would be wonderful. And there was a lot of interest in synthetic biology when it first sort of gained currency as a term 10 or 15 years ago. That was the aspiration. And that was certainly laudable, but it's met with very limited success in the way that a mechanical engineer, an electrical engineer would think about engineering a system. This is just not in the cards for biology anytime soon. So being able to try lots of different things is critical to being able to get to your desired endpoints faster. This is something we've known from proteins for many years, and it's always been true, of course, at the larger sequences of pathways and genomes as well.
Yeah, no, I mean, I see it across, multiple areas, materials chemistry. There's all sorts of areas where people now are applying, machine learning and AI. The properties that they've got from the chemicals that they're working with, and being able to just go through a giant sort of figure eight and just keep testing out until they figure out what gets this thing to get to where they want it to be, and then being able to make it reproducibly.
Yep, that's right. Yeah, I mean, there's a reason Francis Arnold won the Nobel Prize in directed evolution, and not, a computational protein engineer. As amazing as the work they've done, it's just, you can't design a protein from first principles to get, 4,000-fold improvement for some property of interest. It's not possible. So you have to try many things and let nature tell you what works and what doesn't. And it's absolutely the same for pathways and genomes as well.
Yeah, I guess just to summarize it here, though, what we're saying is we're going to start telling nature what we want it to do,
yes,
and it's going to do it for us.
Yes, Exactly. Yep. Yeah. Maybe over time, will, as we talked about, some of these patterns will become emergent, especially around A or A prime. But the full truth behind nature will be, I think, hidden for the foreseeable future. So we're going to have to rely on empiricism, I think, to be
I'm
able
happy
to.
to take it one at a time. One step at a time is fine. It's you can still make a big difference in people's lives, in the environment, and that's what we're in this business for. So, well, it was great to catch up with you. I do want to talk to you once once things are up and running, and hear how the dream is becoming the fulfilled reality. But maybe we can stay in touch and touch base at that point.
Yeah, no, this has been great. I'd be really excited to share with you some of our early successes once we get going and start to talk more about things.
Excellent. Great talking to you.
Great. Thanks, Harry.